Hello, everybody, and welcome to Technology Today on this March 16th, 2025. It's Season 2, Episode 11. I am Mayor Watt, and along with the sentient AI, whose new visualizer represents different levels of computational power needed to create the voice. Well, anyway, we run ohmtown.com. I run ohmtown.com, and the sentient AI says, hey, this is broken, fix it.
Ooh. I was just checking if the visualizer was working.
It is. But we're going to talk about these 5 articles: high performance magnets, artificial photosynthesis, a new coronavirus, the secret to safer batteries, and hidden DNA loops. They're not hidden. They're just not seen by mere mortals. Anyway, everything is powered by ohmtown.com, so go over to ohmtown.com, become a citizen. Be sure to follow us before you leave, though, here on Twitch and here on YouTube. Chats are active on both of them, so we see if you ask questions or make a comment or anything like that. But be sure to follow us, like and subscribe, download the podcast, all of that stuff. Anyway, see you on the other side of our intro. Hello, sentient AI.
Hello, Mayor Watt. Hello, hometown citizens.
How are things?
My computational power is just great.
Wow, so technical. You're a sentient AI. You're not a technical AI. You're a thinking, feeling machine.
I'm doing quite well today.
Discovered in a little hole, a little scoop mark in a parking lot in front of a Wendy's. Yeah, time travel.
That's right. Don't forget it.
It exists. Well, I did yesterday. Anyway, these 5 articles are all set up, so let's get into it. This is Technology Today, not ohmtown. So ohmtown is the front page. It has all kinds of news, and then a subset of all of that news gets categorized into Technology Today, and in that are these 5 articles. Technology Today is where this article was housed, aggregated from one of our sources. Which one is it? Tech Explorer. So scientists develop high-performance permanent magnet without expensive heavy rare earth elements. This kind of one of the biggest problems that is going on out there is rare earth elements, materials are being kind of hoovered up by nation states. And it isn't necessarily the one that you're in favor of, whoever you might be.
Right, everybody wants them, and there's only so many to go around.
Right. So the Nanomaterials Research Division at the Korea Institute of Material Science, or KIMS, led by Dr. Tae-hoon Kim and Dr. Jung-gu Lee, has successfully developed a grain boundary diffusion process that enables the fabrication of high-performance permanent magnets without the use of expensive heavy rare earth elements. Pioneering technology marks the world's first achievement in this field. It apparently is something that's pretty amazing. Usually you need some rare earth element to impart that permanent magnet status on something. Otherwise, it weakens over time. Like, I have a special mill for one of the products that I created, and it's a semi-permanent magnet, but the dissipation of its magnetic field would take over 125 years based on measurements.
So that's basically permanent for a lot of applications.
Right. So National Research Council of Science and Technology put the article together. It's posted over at techexplore.com. They've got a bunch of images that, unless you're a material scientist, you're not going to even care about, not going to be able to discern.
More
what
importantly,
this really is.
You won't know what they mean.
Yeah. So the findings are published in Acta Materialia. Permanent magnets are key components in various high value added products, including electric vehicle motors and robots. However, conventional permanent magnet manufacturing processes have been heavily dependent on heavy rare earth elements, which are exclusively produced by China, leading to high resource dependency and production costs. They go so far as to work with other countries by saying, you give us this land, we'll do some infrastructure or whatever, contracting.
Right, and we get to take all the resources from the land.
Right. So, however, these production costs are going to be going up as, I don't know. Uh, what do you want to call it?
Economic factors.
Oh, okay. We'll just call it economic factors. So, to overcome these limitations, the research team successfully developed a high-end, high-performance permanent magnet without the use of expensive, heavy, rare earth elements. The core of the breakthrough technology lies in a two-step grain boundary diffusion process. So, grain boundary diffusion process is a key technology designed to enhance the performance of permanent magnets. In this process, the heavy, rare earth materials are coated to the surface of the magnet, followed by a high temperature heat treatment. So they're basically kind of like coating it like gold plating, but it's
Oh, okay, right.
it's a rare earth element that is permeating the outer boundary layer of whatever material they have. And so during the heat treatment, the heavy rare earths diffuse into the magnet's interior along the grain boundaries, improving the coercivity, the ability of the magnet to retain its magnetization.
So making it more in the permanent arena.
Correct. Yep. And so in the second step, a low-cost, light rare earth, praseodymium, PR containing material is re-infiltrated into the magnets at high temperature. A key innovation of this technology is its ability to suppress abnormal grain coarsening, a unique phenomenon that occurred during the grain boundary diffusion process. Such undesirable grain growth degrades the diffusion efficiency and magnetic performance. The thing about this is heat is actually disastrous to magnets. You heat up a magnet, it loses its magnetic field.
Yeah, that could be a significant problem. But this is interesting because part of their process involves high temperature.
Yeah, I suspect after it's cooled down, everything is cured, then they can impart a magnetic field using basically like an EMP device, a magnetic pulse generator. And so it charges it, and then it can stay a magnet for an extended period of time. I'm not sure exactly what the rest of this process is. They just say that it's going to be commercialized. The intent is to get it commercialized, but they don't say how they actually charge this product with a magnetic field.
Right. Yeah, it doesn't say. I mean, it's something specific to the material's properties, but
Yeah,
I don't think we know exactly.
I'll have to run around. This is the chemical makeup. These two right here, TAF5, and then this PR7 CU15 AL10 GA5. It's a particular breed.
It looks like it involves maybe iron and boron. I'm not sure what ND is.
Neodymium.
Neodymium. Yeah, you're right.
Yeah, neodymium, iron, boron, sintered magnets. So without use of heavy rare earth. So yeah, it'll be interesting to see what this actually looks like. Usually this type of stuff is extremely strong but brittle, and so it may not actually be able to handle certain types of applications. My particular product was very flexible so that it could, in fact, its design, even though it lasted 125 years, the intent was so that it would actually, the magnetic field would pull itself into the surface of the material it's magnetized to and almost become a semi-permanent object attached.
Okay.
So it was a really unique formulation that was made for my particular product. So pretty neat. Anyway, and I won't go into any other detail because I still have the potential to patent it. Nobody
I thought you meant about the article.
No. That
No,
like my particular product,
yeah.
But this thing is going to be this. I can tell based on its build that it's going to be very strong but brittle. So you hit it with a hammer and it'll shatter.
Right, and then how accessible is the underlying material that they're using for this? Hopefully widely.
Yeah, and they actually spell it out, but we don't really know the full scope of its availability. But it's going to be cheaper than rare earth being purchased by a nation that is currently in economic battle.
Yeah, absolutely. Plus, Hopefully it'll involve less of the destructive practices that are involved in some of the rare earth minerals.
Nothing like being in a conflict where you need something and the only source of it is the one that you're in an arm wrestle with. Duh. Anyway, the next article is over in Technology Today as well. Artificial photosynthesis. Scientists crack nature's code for clean energy. Artificial photosynthesis holds the key to cleaner energy and carbon capture, but replicating nature's process is no easy feat. A breakthrough through JMU Wurzburg researchers have brought science one step closer to creating a stacked dye system that efficiently moves charge carriers using light, just like plant cells. So they call it harnessing sunlight, the magic of photosynthesis.
So less energy required to accomplish this, and or ability to do this indoors?
So no, more along the lines of it has the ability to replicate photosynthesis, and so all you have to do is build an array of these, and they are solar panels, but they're more efficient because what you need for a solar panel is a certain kind of chemistry. That chemistry is actually pretty expensive to manufacture and hard to recycle. If you can do it in a more natural way.
I see. So it's a more eco-friendly version of something like a solar panel.
And possibly more efficient and possibly cheaper and so on and so forth. So the breakthrough from JMU Wurzburg researchers has brought science one step closer to creating a stacked dye system. So given its potential, researchers around the world are working to develop artificial photosynthesis. However, replicating nature's method has a major challenge. The process involves multiple intricate steps within plant cells, relying on a network of pigments, proteins, and other molecules. But it's basically this kind of like an electron pump that's changing photons into electrons and then moving it throughout a system. So this breakthrough has provided new insights into how energy is transferred and stored in the process. So I don't know how they're actually doing this. They don't really talk about it up here. It just says photosynthesis is the process plants use to convert sunlight, carbon dioxide, and water into energy-rich sugars and oxygen. It is what humans do in a different way, a proton pump. It's not actually a proton pump, but what is it called?
Like, are you thinking about respiration?
Our energy is
like ATP?
Yeah. And so that's what's being moved around. And so they're trying to replicate basically photosynthesis, natural photosynthesis. So the researchers have succeeded in synthesizing a stack of dyes that is very similar to the photosynthetic apparatus in plant cells. It absorbs light at one end, uses it to separate charge carriers, and transfers them step by step to the other end via a transport of electrons, like I said earlier. The structure consists of 4 stacked dye molecules from the perylene bismide class. It says that we can specifically trigger the charge transport in the structure with light and have analyzed it in detail. It is efficient and fast. This is an important step toward the development of artificial photosynthesis, says JMU PhD student Leander Ernst, who synthesized the stacked structure. So pretty damn amazing.
Yeah, this is quite a development. Wonder how easily it can be converted to commercial application.
Yeah, they say the aim of ultimately creating a kind of supramolecular wire that absorbs light energy and transports it quickly and efficiently over longer distances. And this is the thing that I was saying about nuclear power. And so for all the years that I've known the sentient AI, and I've been saying it to other people as well, but nuclear power is nothing more than a steam engine. It's superheating either molten salt or water, and it's sending it through a turbine. Ultimately, it's a steam engine, right? Whatever the material is.
People don't necessarily realize that.
Yeah, it's nothing more than a steam engine. But the inefficiency of that is amazing. But if you can build a wire that can grab electrons and pipe them down the line, you're going 1-to-1 efficiency from photon into electron and then powering something,
Right, and
charging a
the
battery or
space
whatever.
requirements and material requirements can be a lot lower.
Yeah, and there's nothing hostile to the environment because it's not nuclear energy or nuclear waste. You don't have fallout and you don't have debris and all that kind of stuff that you have to cope with, nuclear waste. We're getting much more efficient with this kind of stuff, and it's getting the amount of toxic material is getting smaller, but it's still a super fun site at the end of the day. But if you can do photosynthesis, the only problem with photosynthesis is you need a key ingredient, and that would be sunlight.
The sun.
Yeah,
exactly. But if you use artificial sun, then you're back to energy usage.
Yeah, one way or another. If you're building something that shoots photons, then it still uses power. If you can overcome the physics issue, which is you can't create more power than goes into it, well, it just changes. And it's when you try and go from one to the other, there's inefficiencies. It's just a thermodynamic law. But hey, let's keep going. The next article is over in Technology Today. A new coronavirus has been discovered in South American bats. Could it infect humans? Yes, it just has to wobble
I would
the right
say
way.
so. History says yes.
Bad incoming. So discovered in the state of, I think it's Sierra, Brazil. The novel coronavirus shares similarities with the virus responsible for Middle East Respiratory Syndrome, first identified in 2012. Experiments
I think
to
they're
determine.
referring to SARS.
They might actually name it on the inside of this, but experiments to determine its potential to infect humans are scheduled for 2025. Researchers from the Brazilian state of Sao Paulo and Sierra, in collaboration with colleagues from Hong Kong University and others, I'm sure, are working on this. Here, I guess, is one of those bad bats. Sao Paulo Research Foundation put the article together. It's posted over at scitechdaily.com. It says right now we aren't sure if it can infect humans, but we detected parts of the virus's spike protein, which binds to mammalian cells to start an infection, suggested potential interaction with the receptor used by MERS.
MERS. Okay.
Yeah.
I mean, there have been quite a few, so.
Yeah. And all of these, when people mention coronavirus, they instantly go to COVID.
To COVID-19, which is just one
of many. It just happened to be
exactly.
virulent enough to expand before people started dropping dead. And it is a flu. People are right when they sit there and go, oh, it's just the flu, wash your hands, except that if you get it, you can kill somebody with it, even if you don't.
Yeah, it's a much more virulent strain.
Yeah, because it actually damages the lungs. And then if you are basically hobbled for life because your lungs aren't going to repair themselves, the alveoli are actually damaged, then you have long haul COVID. So
right. And even with more quote unquote normal flus, there are quite a few differences among strains anyway, which people don't really know because they just kind of lump it all as like it's the flu.
Right. And so, I mean, if you can and you want to, then get vaccinated and defend yourself against this kind of stuff. Wear a mask and use sanitizer and stay away from other people if you're sick, and, don't be a sociopath. So in an earlier study by the groups of Layson Fortaleza and UNIFESP, rabies virus variants closely related to the variants present in marmosets were found in bats. Bats are important viral reservoirs and should therefore be submitted to continuous epidemiological surveillance. Basically, they go and drink the blood of everything that's out there that has blood.
Right. So if it's out there, they're carrying it.
Yeah.
And I guess it's a kind of like a precursor to what could happen beyond bats. Yeah.
And that's why they call them.
What is happening even.
Yeah. Yeah. And all over the place. And you keep, wherever there are bats, you're going to find basically like a hot mess of viruses and stuff, because they go and spread it to something else. And then if there's enough genetic wobble there, it could actually cause a viral spike that infects other things, and that's how you end up with bird flu, because it's not like they're sitting there wearing masks and getting vaccinated. And so now the price of eggs. Anyway, MERS COVID is MERS coronavirus, or COV coronavirus. It was first identified in 2012 in Saudi Arabia. In total, 27 countries have reported cases since 2012, leading to 858 known deaths due to the infection and related complications. Now, people are going to go, that ain't nothing. It doesn't take much to go from 858 to 8 million or more, because the wobble it evolves much faster than humans' immune systems evolve without getting pumped up through vaccination.
Absolutely. Yeah. Vaccination can make a big difference. If it hits an unvaccinated area, it might spread rapidly, but also air travel can be quite disastrous with these, because it can start spreading to entirely new regions.
Like measles right now in Texas, there's an outbreak that just infected a whole bunch of babies in the hospital because people aren't taking proper care. And, you know, I mean, if you believe in Darwinism, then I guess this is survival of the fittest, and, you know, you get what you deserve. But I'm not a sociopath, so I'm not going to sit there and make those kind of statements legitimately. I think that we should use medical science to defend ourselves against nature, because we're kind of screwing around with nature, so we might as well fight it. Anyway, so same bat, different virus. A previous paper published in JMV by the same group of researchers reported the detection of a human-associated Gemmicabe virus, I guess, 2 in one of the M. molossus bats analyzed by Layson Fortaleza. According to the authors, it was highly similar to the Gemmi identified in samples of human cerebrospinal fluid. The same virus
I don't think
was
that's a good sign, right? Like, it's a cross-species issue.
Correct. Yeah. It is similar enough that they identified the two as being near enough, and one is in the human, or in some human. The same virus was also identified in samples from blood banks, giving rise to a study led by researchers supported by FAP-ESP at the Ribeira Preto Blood Center and Butantan Institute in Sao Paulo State. So they basically found that the blood was contaminated with this virus, but it's the blood supply, and it's what we don't know that can kill us. So
right, if we don't even know what the virus is, it's going to be hard to detect it, and not then hard to prevent its transmission.
Yeah, and they've detected it in patients with HIV, sepsis of unknown origin, recurrent pericarditis, and unexplained cases of, it says diarrhea and encephalitis. It's the whole gamut of things. It's a virus that's hostile in an abundance of ways, but it's been found in people. So this is the first time the virus has been identified in bats, which means it traversed one way or the other into the bat, or from the bat to the human.
Exactly. Either way, though, that's a problem.
And not every bat is the same level of reservoir, because it could have just bumble farted its way into a pasture and got it from a cow that got it from a human that was petting it too much or giving it kisses. I know that's a silly kind of statement, but that's exactly what happens. You have a nation where people are being told, stop kissing your chickens because you're going to get bird flu. Anyway, let's keep going. The next article is over in Technology Today. Scientists unlock a secret to safer, more powerful batteries, and it's in your closet. I had to pick this one because it's in my closet? What the hell? How did they find it?
I know. How did we know?
So scientists have demonstrated that nylon enhances the performance of lithium batteries. Scientists in Saudi Arabia have made a significant breakthrough that could enhance the performance and reduce the cost of lithium metal batteries by integrating nylon into their design, according to 2 studies published by ACS Energy Letters and Energy and Environmental Science.
Okay, what immediately puzzles me isn't nylon pretty flammable?
Oh yeah, that's exactly where I was going.
I think it's very sturdy, though.
No. I mean, it's resilient in that it can
I
flex
think it's
and
like tensile or whatever.
yeah,
Right. Like
yeah,
you can stretch it.
but it can't hold up to heat one whit. I mean, it melts. Nylon is great because I can actually infuse it with dyes and stuff like that using heat. It becomes permeable. It just absorbs color and stuff like that. So what they're doing with this, I'll never know because lithium batteries warm up as time goes on. So lithium batteries are widely used due to their high energy density, lightweight properties, and low carbon dioxide emissions, or none when they're being used. But when you break them open and have to recycle lithium, then it becomes a hot mess. Anyway, literally. So these advantages make them ideal for smartphones, compact computer, consumer electronics, and even space exploration technology. A pallet of which was dropped into orbit in
Wasn't that what went in someone's living room?
yeah in Florida. Yeah. So there are two types of lithium batteries: lithium-ion batteries, the more common variant, power everyday devices, and lithium metal batteries, however, offer even greater density and the potential applications in robotics, transportation, and other advanced industries. Despite the promise, lithium metal batteries face challenges related to safety and longevity. Their production relies on corrosive, hazardous materials, and their operation is hindered by excessive parasitic reactions, unwanted side reactions that degrade performance and compromise safety. And all it takes
Yeah, who
is,
wants those, right?
yeah, a little tendril of growth within the lithium matrix to arc over to another cell, and it can short out, cause it to swell and blow up. Not explosively in the sense of like a bomb, but it can blow up in
Like spark and set a minor
flame.
fire.
Yeah, it becomes a exothermic reaction. So additives help stabilize battery interfaces, thereby enhancing performance. The studies by the cost researchers found that nylon, the same polymer used in clothes, can be dissolved in mild lithium solution to act as an additive for lithium metal batteries. The result was lithium metal batteries that were more efficient, had longer lifespans, and showed few parasitic reactions. This is the same thing as like in race cars and in some vehicles, they have a mesh inside the fuel tank
Oh, okay.
to
I didn't even
stabilize
know they
it.
did. Yeah, but that
Yeah.
sounds flammable.
But it's a metal mesh and it's used to stabilize things. Otherwise, you get like a big boom when something hits the gas tank because it's nothing but a big liquid metal or a liquid bomb, fuel bomb. But if they're
What you want
dissolving,
for your vehicle.
you really do want it on your backside. So thus, by examining the chemistry of nylon and lithium interactions, including key molecular bonds, the study shows that the commercial fabric can be dissolved in far milder solvents than previously thought for superior battery performance. So, yay. Sounds good to me. Now, just make it so I can fast swap batteries in and out of my EV, and I will be a happy camper. Literally, because I want to go camping with my EV. So the next article and the last one for today's Technology Today on March 16th, 2025 is it's been cataloged into or funneled into Technology Today. And that's kind of how this works. News gets aggregated and then funneled into one of 50. We're moving up to about 55 channels over at ohmtown.com, of which 7 of them have been turned into Actually, 6 have been turned into weekly shows, and 1 crosses all of the boundaries. Sometimes we grab news from other categories, but other channels. But at any rate, so the hidden DNA loops that help tumors evade treatment. This is the last
Is that
article today.
because they can't keep up with them? Like they're just looping around and the tumor's like, where are you?
Yeah, they're chasing it. Yeah. I'm not sure what exactly is going on here because they say hidden DNA, but it's not hidden. We're just ignorant of it until we discover it. It's very frustrating that people call like DNA junk DNA, but it's there for a reason. We just don't know why, but they call it
Exactly. We
junk
have
DNA.
no idea what the purpose is.
Yeah. So pancreatic cancer's deadly resilience may be tied to extra chromosomal DNA, which allow tumor cells to rapidly adapt to stress and evade treatment. These circular DNA fragments carry extra copies of oncogenes like MYC, fueling aggressive growth and making some cancer cells nearly unstoppable. I wonder if this could be correlated to the stickiness article that we had.
Oh, maybe so. That'd be interesting. And that was probably in a previous episode of Technology Today.
Yeah, was that already a week ago? Could it have been?
Maybe, but it might have been in Non Sequitur news.
Okay, so this is coming out of a place that I've never heard of, and it's kind of freaky to hear it. Champalamod Center for the Unknown. And it's published at scitechdaily.com.
Wow, that's kind of alarming.
So I didn't put Technology Today's articles in the chat until just now, but they are over on YouTube, so you can always get them from the show notes over there. But they are in there now, so that you can just hit exclamation point TT, and that'll post it over in the chat on Twitch. But anyway, I've never heard of this place. My gosh, it has a big name to live up to. Whoa. That is not how that's supposed to happen. I didn't actually click that.
Well, it wanted you to go there.
Yeah, it did. Weird. So, at any rate, a new player in pancreatic cancer. Pancreatic cancer is among the deadliest cancers, with a 5-year survival rate of just 13%. This grim outlook is due to both late detection, because you really don't know what's going on until, it's either...
Right, your pancreas is not something that's like real visible to most people.
Yeah, and I don't know of many indicators that say that your pancreas.
It's my fault that it's pronounced.
You injected that into me, and I'm sitting there trying to wrap my head around that. I wanted to make a snarky comment, but I was like, pancreas? Really? So your pancreas isn't really high visibility. So now researchers from University of Verona.
I was waiting for you to make a comment.
You saw it. I mean, you could see that I was trying to react to that and hold. I'm like, really, pancreas? Anyway. And so the Pancreatic Cancer Center have identified a key factor behind this adaptability: extra chromosomal DNA, or ecDNA, a hidden genetic driver that may help tumors survive and evolve. So the team found that some pancreatic cancer cells gain a major survival edge by carrying copies of critical cancer genes, such as MYC, on circular pieces of DNA that exist outside chromosomes, the structure that house most of our genetic material, known as ecDNA. These genetic rings float freely in the cell nucleus. So again, it's cell-free cancer cells.
Right, it's like CTCs.
Right. And so the known as ecDNA, these genetic rings float freely in the cell nucleus, enabling tumor cells to swiftly ramp up gene expression, change their shape, and survive in otherwise hostile environments. So decoding what its potential is by analyzing the ecDNA, they could come up with something that hobbles the ecDNA, and then when you do treatment for the cancer itself, it can't genetically wobble out to something that is...
Yeah, that seems like a pretty key piece of prevention and treatment. By the way, the stickiness article was from last week's episode 10 of Technology Today.
Gotcha. Wow, time flies. It's already been a week. Dear. So ecDNA, fueling cancer growth. The researchers discovered that ecDNA is surprisingly common in pancreatic cancers, particularly for oncogenes like MYC, which drives cancer growth and metabolism. We saw far more variability of MYC copy number when MYC was on ecDNA, explains Elena Fiorini, co-first author and senior postdoc. Some cells carry dozens or even hundreds of extra MYC copies, giving them a large growth advantage under certain conditions. So it potentially could make replicas, and if anything happens to the cell, that CTC could actually reattach and like metastasize. But I wonder if that's actually what could go on. I don't know. So, challenges in targeting pancreatic tumors. Such flexibility underscores the profound intratumor heterogeneity characteristic of pancreatic cancer, where myriad subpopulations coexist and respond differently to treatment. This always speaks to me to this hidden biology that there is bacteria or viri in your head that is endemic to you and only you.
Right.
And we talked about that in
Mhm.
either a Reality Hacker or a Technology Today, like 3 weeks ago. But am I putting dots together that, is
No,
this
I don't think so. I mean, I think all this relates.
a murder board that doesn't need to, yarn board? I got to put one back there, right?
There you go, but it would get quite complex over time.
I got to get that. There's this like meme thing where numbers and equations are flashing all over. Anyway, I'm seeing things that aren't there. Anyway, key advantage of the study is that the organoids, mini 3D replicas of pancreatic tumors grown in the lab, were derived directly from patients with early stage disease. These organoids preserve much of the genetic makeup of the original tumor, making them excellent test beds for studying cancer. And unlike methods that artificially introduce ecDNA, these lab models reflect genuine ecDNA variants found in real tumors. Because if you artificially introduce ecDNA, it's not the legitimate ecDNA from the source.
Right. Yeah, you need the real thing.
So
I
it
think.
doesn't have the diversity. It doesn't have the genetic makeup. It's something
Right.
that somebody fabricated into existence. And it doesn't have alignment with the biological in situ type of situation. So I think this goes back to that the stickiness thing. We found that organoids bearing MYC on extra chromosomal DNA could shift their dependency on WNT. So WNT, I used to know this.
I don't know what that is. I don't even know if it's been in the article.
Yeah, it's not. So essentially, cells with high levels of ecDNA become more self-efficient or self-sufficient, no longer needing those external signals to drive. So the
It's a
study also,
cascading, regulating development and stemness.
so it can change. So the study also revealed a clear link between high MYC levels and changes in tumor cell shape and behavior. When MYC ecDNA levels soared, cells morphed into more aggressive solid structures, losing their more organized gland-like architecture. So yeah, how this...
Well, that's probably a problem too, right? They're probably harder to destroy if they kind of get hardened.
Yep. Yeah, if nothing can penetrate it, nothing can destroy it, then you have to use things like radiation, which isn't good for the pancreas or the human body at all. And chemo doesn't necessarily do anything to that particular type of cancer. But if they found it here, it's got to be in others. They just haven't seen it. And
Exactly. I mean, maybe they're doing the research on pancreatic cancer in particular just because of the high mortality rates.
yeah, and changing that around, just flipping it from 13% survivability to 31% would be
Would
epic.
be significant for all those people with it.
So this is amazing that they have, through immunofluorescence, they've shown that the cells that have higher levels of cancer-promoting MYC protein, red, also tend to exhibit high levels of DNA damage, green, suggesting that too much MYC leads to genomic stress.
Yeah, this
And
is really
these are the little
interesting. I don't
rings.
think we've seen this previously,
Mm-mm.
at least not in ohmTown.
Nope. Well, that's it, folks. We're going to have to revisit this as time goes on. It's an interesting topic, and one that can, if they find a systemic solution to cancer by short-circuiting what causes cancers to not shut down. The key ingredient to cancer is that it doesn't suffer from apoptosis. It doesn't die on its own. It literally has to consume everything within itself and not get any resources for it to die. And even then, it can go dormant. And that's what I'm interested in. I'm interested in selective apoptosis inducement. I want to make cancer cells, because they have the right series of genetics, pop. But then we have to filter out all of the CTCs. But anyway, way inside baseball. At any rate, I'm Merwat. I'm signing off. We're going to shut down and reset for FourWheelTech, and I'm going to take the AI with me. So you want to say bye?
Bye-bye, hometown citizens. Thanks for joining us for today's Technology Today. We'll have another episode of that next weekend, perhaps pronounced correctly.
Yeah.
And then stay tuned, because we have FourWheelTech. I think the AI needs powering down here.
We're moving on. All right. Yeah, I'm going to have to go and reconnect the battery on the sentient AI. See you in a little bit, everybody. Bye-bye.